Welcome to Unlocking the Mysteries of Cell Division, your ultimate guide to understanding and fascinating processes that drive life itself. In this introductory episode, we will embark on a journey into the intricate world of cell cycle, exploring its phases, regulatory mechanisms, and real-life implications. Join us as we unlock the mysteries of cell division and discover the fundamental processes that shape the world of biology. Whether you are a curious learner or a seasoned scientist, this series promises to illuminate the pathway toward a deeper understanding of cellular dynamics. So without further ado, let's dive into the captivating realm of cell division. First of all, when we say cell, this is the structural and functional unit of life. A lifespan of the cell is what we call the cell cycle. And I need you to understand that the lifespan of the cell can vary greatly depending on the types of the cell and the organism it belongs to. Some cells, like those in human body, can last for days, months, or even years, while others, such as the certain immune cells, have shorter lifespan measured in hours or weeks. There are four main phases of cell. We have the G1 or G phase, G1 phase, which is also known as GAP1. We have the S phase, known as synthesis. We have the G2 phase, which is GAP2, and we have the M phase, which is mitotic. And we also have the interphase. When we said interphase, this is simply the collective term of G1, G2 synthesis, mitotic phases. Interphase is the longest phase of the cell cycle, during which the cell grows, replicate its DNA, and prepare for cell division. Also, when we said interphase, this is simply, we can just put it differently by saying interphase is simply the longest phase of the cell cycle while the cell prepares for its division. From our diagram, here, this is our interphase. So in this interphase, it consists of the G1, and in this G1, which is the G phase, the cell grows here and carries out its normal function. It prepares for DNA replication. DNA replication actually happens in S phase, which is for synthesis. Specifically, the synthesis is for DNA synthesis. So what happens next? The most cells live here, that is in the interphase, except for the cancer cell. And another main phases of the cell here we have the active cell division. When we say the active cell division, this is also known as the mitotic. Active cell division, which is also known as the mitotic. It also includes two main processes. We first of all have the mitosis, which is this, and we also have the cytokinesis. When we say mitosis, this is simply the nuclear division. And we say the cytokinesis, this is simply the cytoplasm or division of the cytoplasm. Other part of the G interphase are G1. We say G1, and we call G1 to be the longest part of the cell. And in this G1, most of the cell life spent it here. Let's write it down. The cell growth and carries it. The cell grows here and carries out its normal function. So next thing we have on the list is the S phase. So when the cell moves from G1, actually, it moves to the direction of the S phase. From G1, because from G1, the cell has a choice to continue growing. If it wants to move, it moves to the direction of the division cell. Meaning from G1 to S phase, from S phase it moves to G2, from G2 it moves to M phase, and that's how it continues. So what I'm trying to say, from G1 cell have its choice to continue growing. If it wants to move onto the direction of the cell division, it moves to the next phase called the S phase, known as the synthesis, typically for DNA synthesis. What happens in the S phase? When the cell moves there, it takes 6 to 8 hours for DNA replication. They replicate and end up becoming 46 chromosomes. In this case, the cell will continue to grow and prepare for next division, because from the S phase, it's also going to continue moving until it reaches the G2. So in this G2, which is here, how long does it take? It takes 4 to 6 hours. So let's take a quick revision on the S phase. In the S phase, we said the DNA replication occurs, resulting in the synthesis of identical copies of the cell genetic material outside the chromosomes. Here we're going to have 23 pairs of chromosomes taken from both parents. 23 multiplied by 2, this is simply going to be 46. In the G2, what happened? It takes 4 to 6 hours. Cell continues to grow and prepare for cell division. We said the cell continues to grow and prepare for the mitosis. It synthesizes protein and organelles needed for the cell division. That's what happened here. So when it moves from G2, next phase is what? Is M, which is known as the mitosis. And we say the mitosis or the active cell division is being divided into two. We have the mitosis and the cytokinesis. The mitosis is also being divided into 4 main divisions, which is the telophase, anaphase, metaphase, and prophase. The mitosis is the process by which the duplication of chromosomes are separated into 2 identical sets, each going to one of the daughter cells. It division into 4 stages, which we said the prophase, metaphase, anaphase, and telophase. Let's look at it one by one. What happened in this prophase? In this case, the first thing that happened in the prophase, the chromosome condensed and become visible under a microscope. The nuclear envelope dissolve and spindle fibers. Next thing that happens is on the chromosomes, metaphase. In this case, chromosomes line up at the metaphase plate, a plate equidistant from the 2 poles of the cell. Next thing is anaphase. What happened in the anaphase stage? Sister chromatids separate and move toward the opposite pole of the cell, pulled by the spindle fiber. Next thing we have is the telophase. The chromosome decondensed and nuclear envelopes reform around state of the chromosomes. The cell prepare to for cytokinesis. From the telophase, it moved toward cytokinesis. When we say cytokinesis, this is simply the final state of the cell division, which is this. What happened at the kind of cytokinesis? This is the place where the cytoplasm and organelles are divided between 2 daughter cells. In animals, the cytokinesis involves the formation of cleavage furrow, which pinches the cell membrane inward until the cell divided into 2. In plant cells, a cell plate forms between the 2 daughter nuclei, eventually developing into a new cell wall and separate the daughter cells. So that is what happened in the cell process. We can finally summarize everything by saying the cell division is tightly regulated to ensure accurate duplication and distribution of the genetic material to the daughter cell. Regulatory mechanisms such as the checkpoint, cyclins, and cyclin dependent of kinases, which is CDKs, ensure that the cell progression through the cell cycle is coordinated manner and the errors are corrected promptly. Understanding the cell division is essential various fields in biology, including development of genetic and medicine, as the dysregulation of the cell cycle can lead disease such as cancer. So this is it for this video. Thank you for watching. See you next time.